Hi, holobionts. I am Anjali Rana, a PhD student from IISM Mohali, and I submitted my thesis just 1 month ago. The title of which is Disentangling a Holobiont: Role of Microbes in the Ecology and Evolution of Jewel Wasp Nasonia. Because every multicellular organism, like humans, we do not exist in isolation. Instead, we coexist with trillions of microbes inside us, which is called our microbiota, which mainly includes bacteria, viruses, and fungi. Just like us, there is a tiny insect called Nasonia, which is smaller than our fingernail. It also hosts a community of microbes. So I have studied their microbiome, basically the genetic material of their microbiota, using third generation sequencing technology. Nasonia is a parasitoid wasp, which means it lives and develops inside a fly host. Nasonia genus has 4 species, and I have studied the microbiota of all these species. For the methodology, I have used both methods, molecular methods and cultural methods, to characterize their microbiota. So coming on to the results, first I found that Nasonia microbiota is species specific, which means each species has its unique composition of microbes, and the host phylogeny does not predict their microbiota composition. Also, the microbiota of these wasps is different from their host in which they develop. Second, when I compared the microbiota of Nasonia with other hymenopteran insects, I found that Nasonia maintains a lineage specific microbiota. Third, Wolbachia, which is an endosymbiont and also reproductive manipulator of insects, alters the microbiota composition of Nasonia. As you can see, the microbiota of Wolbachia cured Nasonia is dominated by Providentia, the blue colored one, while in presence of Wolbachia, Myroids is the dominated one, indicated by the gray color. Not only this, Wolbachia also disrupts the lineage specific microbiota of Nasonia. Now we know that Wolbachia is altering the abundance of these bacteria, but we don't know how, and we can't culture Wolbachia in a agar plate like E. coli. So I used the in vivo method, where I studied the abundance of these bacteria along with Wolbachia across the development of Nasonia using qPCR. And I found that in absence of Wolbachia, there are no significant negative correlations among these bacteria. However, Wolbachia exhibits significant negative correlations with two of the most dominant bacteria, suggesting that Wolbachia interacts negatively with the microbiota. Then I wanted to study how these bacteria interact with each other. So I first isolated 18 different bacterial strains and performed their pairwise interaction assays in vitro. I found that most of the bacterial strains from Nasonia, they remain neutral in presence of another bacterial strain, which means they are trying to coexist together, which indicates stability and coexistence of Nasonia microbiota. With this, I want to conclude that this is the first study on any parasitic wasp to characterize the bacterial communities across all species and to investigate in vivo and in vitro bacterial community dynamic. Thank you for listening.